Good morning to everyone. I will start this topic with putting forward two clinical scenarios. This we have to find out intricacies from this two clinical scenario. So let's find out two similarities between two similar scenarios. On the right side, you can see this is a child of 2 little penis, Jews child, with slender body, short stature. You can see different hyperpigmented patches over the mandible and maxilla with hypoplasia of the mandible. This scenario is associated with the important syndrome which is known as Bloom syndrome. Okay. And on the next side, let us correct, right? Let us correct that you all. I think. Yeah. This child is 20 years boy with alopecia scleroderma skin, and the child is having problem with periodic patterns. So the right side, this scenario, in the aspect of this side, you are suffering from Bloom syndrome, and on the left side, it's progeroid. Okay? These two progeroid syndromes are associated with premature aging. So our topic of discussion is mechanism of aging today and some recent updates which are added in the 10th edition of Robbins, which will be dealt today in my lecture. So we should learn definition of aging. What are the theories proposed to explain the aging? Basic mechanism of aging and some important topics which are added in the third chapter. Topics like autophagy, necroptosis, pyroptosis, and let's start with cellular. We should know anything from the definition point of view that what is cellular is a progressive decline of cellular function and viability of particular cell due to genomic instability, which is caused, which is accumulated. All the genetic instabilities are accumulated in the cells due to molecular damage, which are caused by external hazardous stimuli. So many external hazardous stimuli causing different kind of changes of the DNA. These changes are accumulated within the cell, which in turn cause the cumulative effect of decline of normal pathophysiology of the non-physiological character of the cell, which altogether called aging. Now the interesting topic that from single cell amoeba to Homo sapiens, cellular aging is regulated by a set of the genes which are evolutionarily observed. There is no change throughout the evolution of that gene, which causes regulation of cellular aging. Lots of theories are projected to explain the cellular aging, like evolutionary theory, DNA and genetic theory, endocrine theory, free radical theory, mitochondrial decline theory, and cosmetic. But the most important theory we should learn each and every point which is proposed to explain this theory. Obviously, the other theories like free radical theory, mitochondrial decline theory, and cosmetic theory will be there because they are all with DNA and genetic alteration. But this is the most important theory to explain the theory. There are the four basic mechanisms, molecular mechanisms for cellular aging, which are they are first DNA damage. Second, cellular stimulation. Third, defective protein homeostasis, and 4 desegregated nucleic cells. Mediated again, cellular stresses, differently protein homeostasis, and 4 mediated intricacies. And first period. Now previous knowledge from agonism and efficacy that if any adjacent comes from outside, any change is outside, they can cause defined changes and this DNA or that might be a replication of mitochondrial DNA. These changes in the DNA are usually the DNA repair mechanisms. But some changes, like if the base, a huge base is excised or a long strand of DNA is excised, the damage is so severe that it cannot be repaired back to DNA and protein, but back to the normal process. Then the cell undergoes process. But if the cell is not undergoing apoptosis for particular time, after apoptosis হচ্ছে, but we the problems will get at DNA. There are cumulative problems. They are accumulating and causing the decline of this is the various mechanisms. Second, the theory, which is another theory, but it is closely related with the DNA and RNA kinase theory, like free radicals. This is proposed by David Hammond. We all know of organization, there is generation of so many free radicals, like hydroxyl, peroxide, and hydrogen peroxide. They are tablets, right? Some antioxidants like C, vitamin E, you all know from but opportunity establishment is a toxin. No, there are some hazardous still available in the cell and they cause the problem. They cause the damage to the mitochondria and the cell. Okay? So this damage is again accumulating and causing normal. This is proposed by David Hammond. Second mechanism after DNA mechanism. Cellular synthesis. What is cellular synthesis? So each and every cell in our body has a particular replication capability. So after a limited period of replication, the cell went to a stage of non-dividing stage. Okay? This non-dividing stage is known as replicating cell. So before starting to explaining all these things, you have to learn some basic points of cell division. Have you heard the cell cycle before? Have you heard the term? Anyone? Yes? Stand up. What are the stages of cell cycle? Four phases. Maybe C1. Okay. C1. These are theoretically based for synthesis of methods. Like synthesis of what? So during the G1, what is going on? Going on in the G1 process? Protein replications, accumulation of different basic molecules which are different in the DNA, replications like sugar, nucleoside, phosphate receptor, okay? And accumulation of important enzymes which are different from the DNA synthesis. And what is the role of gene nuclease? Same. Accumulation of different enzymes which are different from the mitosis. So these stages are basically controlled by two things: one, cycling, other cycling. Cycling is a protein which is tightly regulated by an enzyme kinase. So kinase, the term means this enzyme works in what? Phosphorylation of particular protein. So when this cycling binds to the cycling dependent kinase, there is no missionary process of phosphorylation of some protein. There are four cyclins. Cycling B, E, A, beta. They are specific to each and every step of the right G1, E. There is a competition Next, G2. Specific for cycling A and cycling B action. So there is basically a transition phase between G1 and G2 and G2. You just want to do this. So important change. Why is G1 and G1 is G2 in? G1 is please stand up. Why is there if there is no transition, the cell will not arrest in G1. So if there is important moieties which are required for the synthesis of DNA or mitosis, if there is lacking of enzymatic stimulants and still the cell is going in, there is transition. The cell goes on the case of synthesis. So there will be inadequate amount of the DNA which are attached because they are defective. The accumulation of defective DNA and defective nuclear protein within the cell, they will cause dysregulated multiple type of growth. The cell of that particular group, the cells which are growing faster having abnormal DNA. So they unregulated abnormal DNA growth causing cancer. So if there is no checkpoint, the cell goes in a phase of neoplastic change. Now, these cycles are also going to be cycles like B, 4 and 6, 2 minus 3, and E minus 3, CDK2, 17, A minus 3, CDK2, and also B minus 1. Now, you can see that the G1 cycle 3 minus 2 the cycle independent kinase 4 and 6 and acting in this. and they are acting in that transition space. G1 and next checkpoint. G2, these stages may be represented by the cyclic along with cyclic dependent kinase 2 and cyclic 3 with if the cell sensing that the cell should go to the synthesis phase of G1 from G1 to S phase, the cell is arrested in is that not dividing state. কোথায় কোথায় পৌঁছায় এবং যারা সেলগুলো টার্মিনাল ডিফারেন্সিয়েশনের প্রক্রিয়া এবং সেগুলো ডিভাইডিং হচ্ছে না cardiac cell, skeletal cell, renal cell. Why this transition is not happening? Because the important is transition. Radiocustomer gene is the law of number 15. In which radiocustomer there is a termination factor known as elongation. How this elongation factor working? Transcription of how this factor is working by where this elongation factor is binding. এটাই তো জিনিস বলছি ভাইটাল সিতে ছিল ভাইটাল সি ভুলে গেলে আমার প্রাথমিক পরীক্ষা দাম হবে না ফিজিওলজি ভুলে গেলে আর হবে না তুমি একবার শিখেছো তোমাকে আলটিমেট রিপোর্ট দিয়ে তোমাদের মনে রাখতে হবে কি ছিল কি কথা বলছো কি বলতে না লোকেশন একটা টু কোথায় কিছু তো প্রোটোটাইপিং আছে এক্স্যাক্টলি So, when a particle is getting synthesized, the cell may go into a stage of G1 to S that has been simplified this as is not inter, the cell is not arrested in G1 to S. Into a fine with in this when it is inactive, when it is active, it should be free. It should be free. There should be any binding with the protein. But when it is protein with the nuclear factor, they cannot bind. So there will be no transcription. So in retroplasma protein nuclear factor is the bind করে থাকে তাহলে nuclear factor reactive হয়ে যাবে কখন retroplasma hypophosphorylated state থাকে hypophosphorylated state in the retroplasma gene hypophosphorylated state they produce retroplasma protein. Now think cyclic it is a kinase which is causing phosphorylation of the protein. They causing the phosphorylation of retroplasma. That means it gets phosphorylated and it gets inactive. So this mediating plasma is free. Now midway is active on that is it can bind in the promoter region and it can synthesize the protein. So this transition is very good in the stage of with that background যদি cell cannot go, that cannot pass that median or this till point, the cell will be stage of G0. This is okay, and aging is associated with progressive replication. There are two mechanisms of the cell can go into a replicating cellular stage. One is telomere accretion, and second is activation of human suppressor gene. This retinoblastoma gene is a human suppressor gene. Can you get another very common associated with the life-threatening syndrome? Life-threatening syndrome P50. So there are two human suppressor genes. Telomere accretion. What is telomere? In a particular chromosome at the end of associated with a long array of PTA, GGGP, there is a single nucleotide overlap at the G prime end, and there is a binding protein, DSP binding protein. Last telomere, it ensures the normal replicative functions. Any গুলো প্রত্যেকটা ক্রোমোসোমের এন্ডগুলো they are skinny. If they bind with each other, they will form a round cell. During the mitosis, the chromosome should be opened up. If there is formation of recombination, end recombination, the cell cannot go into that mitosis. The cell cannot undergo mitosis. So this telomere is binding at the cap, that is at the end of the chromosome. They prevent this recombination. Now, at the end of the chromatin, there is a special nucleoprotein known as telomere, which with each and every process, after a single cell undergoes this similar cycle mechanism, the cell going in a stage where the cell cannot divide further, because with each and every cell cycle, the length of the telomere gets shortened. Each telomere at a function this is known as telomere accretion, and at a particular length of telomere, the cell cannot divide further. This length is known as affinity. Now, what is the role of telomerase? Telomerase is a DNA molecule. They produce that PTA-GGG. That means particular length which are getting short at each and every cell cycle is added by telomerase. About how, like these are the eggs. These are the eggs of chromatins. And if you focus on the whitish, এখানে normal dividing cell এর মধ্যে telomerase activity খুব কম থাকে, যার জন্য after dividing the cell undergoes synthesis. But if there is hyperactivity of telomerase, the telomere cannot be short, so the cell can undergo innumerable number of replications. So there will be overgrowth of a particular tissue. Overgrowth of particular tissue. So if the activity of telomerase within the cell increases, there is chance of neoplasia. Exactly. এটাই বলছে যে প্রত্যেকটা সেলুলার যখন টার্নওভার হয় বা এপিথেলিয়াল রিমুভাল হয় তখন কি হয় telomerase accretion হয়। But if suppose at the time of telomerase accretion there are so many damages occurring within the DNA, that means within the chromosome there might be duplication, deletion, etc., and even break, breach, cell defective. That defective cells are associated with different number of the chromosomes present within the the basic foundation mechanism of neuroplasticity. One of the foundation mechanisms. You should thoroughly understand this concept. Okay? Next, inactivation, activation of the neural suppressor gene. Normally the cell is going in a stage of senescence, there should be activation of neural suppressor gene. Radioblastoma gene as a neural suppressor gene. If it is get activated, activation of radioblastoma occurs in what and how? If there is any cellular damage, radioblastoma gene gets hypophosphorylated. It binds to the elongation factor 2 and elongation factor cannot be promoter activity. So there is no cellular. This radioblastoma gene is negatively controlled. Clear for it. বুঝতে পারছেন কি প্রবলেম? This radioblastoma gene is tightly controlled by T16 and NK4A. Perfect. কি করে? They bind with C, cyclic B, C, K, and 6 and inactive makes that machinery is inactive. This machinery becomes inactive means they cannot bind to the so radioblastoma is activated. Elongation factors are not used. So there is no such expression of protein is in and therefore it protects the cell from unwanted microgenic signal. If there is any microgenic signal, if there is any signal which is causing the cell to replicate, if the cell in anabolic state, যখন সেলের গ্রোথ প্রপোর্শন হয়, under the effect of growth hormone, ESP, etc., which was in the cell to grow in anabolic state, তখন যে বিন্যাস আসে তাদেরকে বলা হয় মাইক্রোজেনিক সিগন্যাল। আমি আসবো পরে। When this microgenic signal is coming, it might activate. So, it prevents. To make I was using inactivation of cyclic B, cyclic 4. Normal radioblastoma gene binds with elongation factor. So if it binds with the elongation factor 2, the elongation factor will not be active. This elongation factor 2 actually gets activated by cyclin B, CTK4. So if this is inactive, how can thus, if there is excessive androgenic signal that will activate the C69 system, which ultimately causes the activation of the radioblastoma, and unwanted signal cannot enhance the cellular. How clear concept? Third mechanism of cellular effective protein. So I think all of you have Pharmacology chapter. So tell me what is chapter? Anyone first tell me what is chapter? Please stand up. These are hits of protein. HSP 70, HSP 5, there are so many 7, 8 classes of chapters. they are extra protein. They are active in stressful condition. What are the normal action? They can cause normal protein folding. Protein structure action. What are the important structures of protein? Primary, secondary, tertiary structure. Protein folding কখন আমরা পাই? কখন হয়? Protein folding is occurring at what stage? At the secondary. So normal protein folding has a mechanism associated with activation of, the normal function of chapter is protein causing the normal protein folding. And also that if the protein is misfolded, this misfolding of the protein gets damaged by two mechanisms. One, autophagy. And second is ubiquity proteasome block. By these two mechanisms, the unfolded proteins gets degraded. Another important function is protein translocation and signal transduction. So, with increase of age, normal protein folding is numbered and more accumulation of unfolded protein. And with increase of age, the normal activity of chapter declines. So, this misfolded protein will be in the cell and causing triggering of apoptosis. If the cell is not dying that particular time, there will be declining normal function. So, there will be similarity. Like dynamic differentiated cell to project, myocardium project, neural tissue project. So, if the chapter gets inactive on that particular area, it may lead to several diseases like degenerative neuropathy, congenital cancer. And fourth important mechanism, underlying mechanism is abnormality in nucleus sensing. At a bottom is the mitochondrial signal. So there are two neurohumoral pathways by which less calorie intake causing higher toxicity. Less calorie paradoxical thing. আমরা মনে করতাম বেশি খেলে বেশি সুস্থ থাকবো। Not how they are working. They are working in two different pathways. Insulin-like growth factor one pathway and second is SART is physiology আমরা বলতে শিখেছি। তোমরাই বলবে SART। Insulin-like growth factor এটা বুঝে নিও। Each chapter task is to molecular level. It will take some time. Okay? ये ले करी। Important mitochondrial signal by which they act over the cell. This mitochondrial signal is known as insulin-like growth factor. They mimic insulin-like growth factor. They mimic the action of insulin. So, in the downstream, they produce activation of two pathways. One, endokinase pathway. Another, AKT kinase pathway and endokinase pathway. Actually, পাঁচটি বলবো না। তোমরা যখন সাবসিকুয়েন্ট নিউক্লিয়াসের চ্যাপ্টারে আসবে বা হরমোনাল চ্যাপ্টারে আসবে, তোমরা অ্যাকচুয়ালি পড়ে বসো। এন্ডো পাথওয়ে নাকি Anyway, these two pathways are activated by the action of insulin and insulin-like growth factor. So ultimately, they causes the promotion of anabolic state and cell growth. So these are called mitochondria. Another thing is startings এই পাতা এখন বললাম second হচ্ছে startings startings are nothing but important engine these are innately dependent protein deacetylase they are active in deprived cells means when the cell is undergoing atomic changes or cell is deprived from nutrient they get active কি করে they inhibit atomic cell apoptosis most common startings সেই apoptosisটাকে undead also they promote the metabolic activity stimulate the protein folding because যদি protein folding না হয় যদি unfolded protein accumulate করে the cell will go may undergo apoptosis of cell so ultimately the startings and also they inhibit the harmful effect of the pre-membrane so ultimately the action of the startings are causing less damage to the cell so if the startings levels are high the cell will not go in a stage of cellular so less calorie intake is causing two things decreased activation of insulin like growth factor related to pathway which causes decreased microgenic signal so cells are not going on the stage of repetitive replication that's what happens when the cell is not going on the stage of cellular synthesis and also particularly cells are intrinsic actually they causing the decreased decline in the apoptosis so less calorie intake is associated with the longer activity and remember rate maintain exercise छोटा छोटा করে নাও anyway এটা গেল DNA and genetic theory another theory is mitochondrial decline mitochondria power of organelles they not only produce ATP also produce some important nutrients like acetyl-L-carnitine vitamin B12 vitamin but these nutrients if given outside they can protect the mitochondria generally cause the cell issues for the target so it is scientifically proven and experimental still this experimental level that acetyl-L-carnitine and hydrogen can cause longer activity of last theory related to the activity is cross-linking theory glycosylation theory HbA1c what is HbA1c actually glycosylated কি হয় please actually চলছে এখানে তোমরা বুঝতে পারছো বলো কি হয় সেই প্রোডাক্টে বা গ্লাইকোসাইলেটে any protein if is non-hexagonally conjugated in the sugar they causing the accumulation of glycosylated product outside the cell so normal nutrients that are created within the cell so cell gets damaged that's why the diabetes is viewed as accelerated aging state and other important disease where these accelerated aging are indicated for the pathogenesis like senile cataract where and other phytoderma that means appearance of tough leathery skin here the main mechanism is accumulation of extracellular glycogen so these are basic theories which are put forward to understand the concept of tough munchy but these are basically very foundation of cell biology now the next step actually is an updated the first update is automatic when the cell is in intracellular organelles causing the turnover and this recycle. This things is getting recycled. This is the see. There is a pinching of particular cytosol. That membrane could be from endoplasmic reticulum or cell membrane. This pinching is looking like omega. This is omega shape. That's why it's called omegasm. After this pinching, there is initiation of vesicle formation. It is known as initiation membrane. The other name is phagophore. These membrane encircling their own organelles or misfolded proteins within it. Misfolded proteins within it. See, it is known as autophagocytes. They form a cargo with intracellular cargo within which the cells intracellular organelles are getting trapped, getting put within that particular intracellular vesicles and they fuse with the lysosomes. These lysosomal enzymes causing ultimately degradation and this process is getting recycled multiple times within the cell. The cell is not damaged, but the cell causing damage and degradation they are own organelles and misfolded proteins. These are tightly controlled by a set of genes known as autophagy related genes known as ATGs. And see, initially there is a pinching of cytoplasmic membrane. Within the cytoplasmic membrane, there is a glycoprotein known as phosphorylated inflammation. There is phosphorylated inflammation in the cytoplasmic membrane, which gets attached with the microtubule related protein known as microtubule associated protein ligands 3. This conjugation is like ubiquitin like conjugation, non-enzymatic conjugation. It causing initiation of phagophore. Initiation of phagophore. So phosphodiamide is P conjugated with glycated C3. This PEC3 conjugated moiety is the marker of the marker of apoptosis. What is the marker of apoptosis? Marker of apoptosis. Fine. Like that. It is also not cell. This is a dedicated program because it is tightly controlled by the company. So that's the marker. This is the marker. Profile marker usually seen in an active stage at the period of deprived. What are the roles for clarification of different issues like it might be associated with dysregulated autophagy, might be associated with hyperpopulation. Second, in Alzheimer's disease, the autophagocyte formation is dysregulated autophagocyte formation associated with Alzheimer's disease. তোমরা পড়ে পড়বে tau protein. That tau protein is basically dysregulated product of dysregulated autophagy. Thirdly, degradation of Shigella, one of the important bacteria, because it's the dysentery. Herpes simplex virus or mycobacteria, they are all degraded by the autophagy. So if the normal autophagy in the cell is defective, they cannot be this mechanism, this microbes cannot be degraded properly. Like ATG5 deletion causes increased permeability, increased susceptibility to tuberculosis. And fourth, one of the recent, I mean 2020, this agonism is put forward in the year of 2020 that initiation of inflammatory bowel disease is associated with a single nucleotide change, which is known as single nucleotide polymorphism of important gene known as ATG16. presently of this ATG system is one thing bacteria. That's all related to. Next topic. Let us take the additional topic. Necroptosis. The name suggesting that necroptosis is combination of two things. One necroptosis and second is why program necroptosis. Why? Because morphologically looks similar to necroptosis, but he also says within the cell there is a tissue necrotic factor receptor. Tissue necrotic factor is a cytokines, which is a mediator of active inflammation. ক্লাসে শুরুতেই বলেছি এটা শুধুমাত্র সেলদের চ্যাপ্টার নয়, এখানে নিউক্লিয়াসে আছে অ্যামিনো ইনফরমেশন ফ্যাক্টর। So this tissue necrotic factor is a initiator of factor transformation that binds with tissue necrotic factor alpha. This causing activation of RIG-1 and RIG-3. This is not gaseous dependent, but this is independent, gaseous independent path. This activated RIG-1 and RIG-3 causing the phosphorylation of MLL, ultimately causing polymer formation. Phosphorylated MLL binds with each other, causing formation within the cytoplasmic membrane, through which the chemokines, cytokines, and different kinds of proteins, cytokines, and chemokines moves outside, and they also recruit the altogether like chemokines, cytokines. These indicators of acute inflammation. So, আমরা বলেছি necrosis acute inflammation হয়, but এখানে যখন necrosis হচ্ছে তাহলে সিমিলার morphological necrosis এর মতন মনে লাগছে because they are associated with the acute inflammation. So they both have inflammatory response. That's why it is called necrosis. The role of necrosis in physiological condition. Mammalian growth plate যখন normal development হয়। Secondly, cell dividing systemic partition includes steatohepatitis, acute pancreatitis, and Parkinson's disease, Parkinson's disease. These are all dysregulated necrosis. Fever, which inflammatory cytokine causing the fever? Interleukin-1, 4. Leukin-1 inflammation starts 6 of them, but mostly interleukin-1. So, predictable toxicity of cells affected by the microbes. They cause the activation of the caspase 1, and this activated caspase 1 converts the inactive interleukin-1 cytokines to active interleukin-1 cytokines, which mediates the inflammation. They also cause the increase in the particles in the cell membrane and all. So this types of cell is associated with micro-affected cells, and this type of cell has last number is ferro means iron, where in this regulated power solution, which is this pathway is depending on iron, because it is seen, it's not totally understood still now, but it is seen that if the iron is low in the cell, this is regulated between power solution and inhibitor. তাহলে আমরা শিখলাম আজকে cellular complex process, cellular integration, complex process, molecular interactions which are seen during the cellular aging process, DNA damage cellular senescence, dysregulated protein homeostasis, and deregulated nucleases. These are four basic. और आपके देखते हैं कि चलना autophagy, genes, necrosis, morphologically, but this program necroptosis. Necroptosis means morphologically like necrosis, but this program autophagy is associated with apoptotic cells and it causes the degradation of all the cells of organelles, and there are some recent updates like complete pathology complete হবে তারও 10 বছর পরে গিয়ে
Pharmacodynamics with understood pharmacodynamics. What is pharmacodynamics? Anyone? Body versus the drug, effect of the drug, and stimulating pharmacodynamics. What is what do we expect? When you are taking a drug, we expect a response in the body. So, response of the drug that we are taking this drug, how we get that response. So, study of biological and physiological effects of the drug on the body or on the microorganisms or parasites in what the body and the relationship between drug concentration and the this is the definition of pharmacodynamics. Got it? Can anyone please break and questions in your paper? Okay. Can you make the definition that is clear? Study of biochemistry and okay. Biological and physiological effects of the drug on the body. Okay. That means a drug cannot give a new biological effect. It can only face up or face down. The effect that the physiological effect already present in the body. Either it can speed up or down or block it. Next, on the microorganisms or parasites. When we are getting infected or infested, that can be affected because it's killing those parasites inside the body, within or out the body, and the mechanism of drug action and the relationship between the drug concentration and the effect. When we are giving a drug, we give it in a specific dosage form. It is going in the body and it is giving a specific concentration. Depending on that concentration, there is an effect. So the relationship between the dose and the effect is also studied in pharmacodynamics. So definition clear? It is either increasing or decreasing the biological action, the parasite microorganisms, or the unwanted cells in the body, like cancer cells. We are killing those, and the effect due to the specific dose, the relationship between the dose and the effect. This is all sum up of pharmacodynamics. Clear definition? Next. So what are the basic principles of drug action? Basic principles. There are 5 basic principles on which a drug causes its effect. First is stimulation. In a drug, it is causing stimulation of a cell and alcohol or retrograde. These are PPI. When we give them, they depress the gastric acid secretion. Gastric acid secretion is normal physiological function. What are you doing? You are decreasing it. It has its own thing. Conduction power. What are you doing? Increasing your pumping activity by giving adrenaline. That is, you are stimulating or increasing your. But you cannot increase or decrease the work and divide the drugs. That this drug will increase, this will decrease. Because there are few drugs which can increase the activity in few organs, which can decrease or repress the activity in others. So there is no clear-cut description that this is a stimulator and this is a depressor. Okay? Depending upon the organs, depending upon the receptors where it is acting, it can either stimulate or it can depress. So if there is no clear-cut demarcation of the drugs. Next, replacement. Sometimes in the body, the organs releases some hormones. In that case, or neurotransmitters released. In that case, if in a very pathological condition, these hormones are decreased. Like in diabetes, what happens? The pancreas is not functioning properly, insulin level is decreased. So glucose level goes up. In that case, what do? You are getting insulin from outside. So that is replacement. A drug can also act as replacement. You are giving hormones because sometimes the hormone level goes down. So you are giving hormones. In that case also there is replacement activity. Cytotoxic action. Penicillin for the gram-positive vaccine. I told you cytotoxic. Cytotoxic is the killing ability. Okay? And without affecting the host cells. That is the cytotoxic action. Irritation. Irritation means it is having a non-specific noxious effect. As a result of that, the cells are getting damaged, there is inflammation, and that is causing the inflammation and thereby the dying of the cells. So that is irritation. So the basic principles of drug action: stimulation, depression, replacement, cytotoxic action, and irritation. Understood how the drug acts? Next, the mechanism of drug action. Two main classifications. Two main broad types: non-receptor mediated and receptor mediated. Okay? So how is the drug acting? It needs something to go and live in the body and get into that, or that is receptor mediated, or else the other part is the non-receptor mediated. What do you mean by the non-receptor mediated? That is by the physical action of the drug. We have seen the physical properties: radiation, osmosis, absorption, bulk portability. These are the physical aspects. So by these actions, a drug can be done. Next is by the chemical action, or chemical reactions in the body. Next, through the enzymes. All biological functions in the body, they are carried out by the help of the enzymes. So a drug can go and through those enzymes, and that enzyme can get stimulated. As you know, these enzymes have either catalytic or the neurons. So what will happen? What will happen? Serotonin level will increase. So serotonin level will increase and that will have an anti-depressant effect. So what is it doing? This drug, SSRI, it is going and binding to the serotonin transporter. What was the work of the transporter? Then the transporter will block the serotonin reuptake. Once the serotonin is released, it is causing its effect. After the effect, it is again reuptake. And the effect then it is gone. But if you stop that reuptake, what will happen? The serotonin will remain there and it will produce prolonged effect. That is the cause of the antidepressant. Okay? Next. Non-mediated by physical action. I told you osmosis. 20% mannitol in cerebral edema. When you give mannitol in cerebral edema, we expect pressure to go down, intracerebral pressure to go down, and that is the osmotic effect of mannitol. Next, absorption, activity control. They were said comes 6 count. You want the mucus to become less thick so that you can expect. So they were said it comes down. Then radioactivity, iron 131, which has radioactivity and it is radioactive, that is hyperthyroidism. Non-receptors will given by chemical action. So physical action we have seen. These are all the physical actions that are producing the action. Now chemical action. Antacid. What is it doing? Antacid. It is neutralizing the gastric acid. Fuming agents like the dimethylchlorine, arsenic poisoning, dextrosolution, iron poisoning, and T-sensitivity in copper poisoning. These are all by chemical reactions. Fuming agents. Okay. Next. Non-receptor mediated enzyme mediation. I told you, drugs increase or decrease the rate of enzymatically mediated reactions. There are biological reactions going on in the body. These are mediated by the enzyme. What will the drug do? Either it will increase the activity or it will decrease the activity of the enzyme. Enzyme stimulation, like adrenaline, increases the hepatic glycogen phosphorylase. Enzyme inhibition. These are all two types. Competitive, non-competitive. Enzyme inhibition means it is blocking the effect. Enzyme inhibition. This you have to understand. Look at the diagram. That is the enzyme. A substrate is there. Inhibitor is there. Substrate normally present in the body. Coming and binding to the enzyme, product is formed. That is producing the normal physiological response. Now, when the inhibitor is coming, what is happening? It is coming to the same site, like the substrate. If it is coming and acting at the same site as that of the substrate, that is the substrate binding site, then it is the competitive. But if this inhibitor is coming and binding to any other site of the substrate binding site, then that is the non-competitive. Okay? Understood? Competitive, non-competitive. Now, competitive is again of two types. Equilibrium, non-equilibrium. What is the equilibrium type? Substrate and inhibitor. They are both trying to bind at the same site. Okay? But if you increase the concentration of this substrate, the substrate will win. It will come and bind at this site and it will overcome the effect of the inhibitor. In that case, that is the equilibrium type. What is non-equilibrium type? If this inhibitor is very strong, now bind to its site and it will produce a permanent damage to this enzyme. You are going on increasing the substrate, but there is no effect. non-competitive. Okay? So competitive, two types, equilibrium, non-equilibrium. In equilibrium type, what happens? If you are increasing the substrate concentration, then the substrate is inhibitor is replaced and efficacy. In the non-equilibrium type, what is happening? It is coming and binding to it permanently. However, you are increasing the substrate, but you cannot get the response anyhow. Okay? The combination of two types, equilibrium, non-equilibrium, and non-competitive, we have next. So these are the examples. See, competitive, enzyme, substrate, inhibitor. Both are competing with each other. Enzyme, inhibitor complex is formed, and this activity is blocked because the inhibitor is going and blocking. It is not allowing the substrate to come. What is happening in the non-competitive type? Enzyme, substrate, inhibitor. The inhibitor is binding to the other side the substrate binding side. So that is the example of. These are the examples of competitive and non-competitive mediating approach. Next. Now next is the ion channel. I told you by the physical effect, by enzymatic effect, then we have some ion channels. Ion channels where we are present. Similarly, ion channels to the fabric. Then different inside outside. Inside. Only cellular ion channels. Both. So what is the word? Transmembrane. That is having an outside part, inside part. So ion channels participate in the transmembrane signaling and regulate the intracellular ions. Intracellular ions factor. Intracellular ions intracellular ions থাকে intracellular ions হচ্ছে কি হয় either cell activated depolarized polarized একটু পড়েছো তো hyperpolarized নালে these ion channels they participate in the transmembrane signaling and regulate the intracellular ionic component either a drug will come and it will bind to a biochemical agent and it will open up the channels that is called a ligand-gated channel. ঠিক আছে? Like nicotinic receptors. Ligand-gated মানে একটা biochemical substance থাকে এসে bind করবে না একটা চ্যানেলে তখন চ্যানেলটা আইদার ওপেন আপ করবে আর ওপেন আপ করে কি করবে ইন্ট্রাসেলুলার থেকে এক্সট্রাসেলুলার করবে বা এক্সট্রাসেলুলার থেকে ইন্ট্রাসেলুলার করবে that is the ligand-gated ion channel. G protein regulated channels. G protein regulated, G protein receptor as a you see the transducer mechanism. Why it is called G protein? Because the cardiac T1 accelerated receptors are G protein regulated. Voltage operated and state sensitive. Local anesthetic of the sodium channels. Depending upon these channels, depending upon the ion concentration, there is voltage. In voltage sensitive, if there is slight change in the voltage, what will happen? The channels will open. That is voltage operated channel. So three types of channels we have. Either a biochemical regulated channel, that is a ligand-gated. Next coupler, G protein coupled. G protein is a transmembrane protein that is present transducer effectively that is non-present, that is the G protein regulated channel, and third voltage sensitive ion channel. Okay. Next mechanism is by the help of receptors. This is a major part of the pharmacodynamics. Right? How a drug is producing a cell. It is mainly with the help of the receptors. What is the receptor? What is the receptor? A macromolecule or binding site that is located on the surface or inside the receptor cell So again, a macromolecule binding site that is located at the surface or inside. Then what will happen? It will recognize the signal or the drug, and it will initiate the response, but itself has no function. Okay, clear? Regulatory macromolecules, mostly they are protein. Sometimes nucleic acid in nature. Mostly these receptors are protein in nature. They are the outer surface of the cell. It can also be inside. But when they are inside the cell, in the nucleic acid, what is it doing? Inside the cell, in the nucleus, then in the nucleic acid, what is it causing? By the gene regulation, either the protein production will increase or it will stop, and in that way it will initiate the response. Okay, when inside the cell, it is in the nucleus, in the nucleic acid gene functions, then what happens? Protein synthesis, either it will increase the protein synthesis and in that way it will initiate the response, or it will stop the protein synthesis and in that way it will block the response. Okay, so this is it is synthesized by the cell and it has definite lifespan. It has definite lifespan, the receptor. Again, new receptors are generated and it will be dead. What are the functions? Recognition and binding. I told you, recognize and bind. Propagation of message. It will give signals. Initiate the response. Then it will give the message. Then we have to do this. Amplify the signal. How amplify? I told you, if it protein synthesis, if it is increased, the response is getting amplified. Integrate various extracellular and intracellular regulatory signals. If a signal is coming extracellularly, that has to go intracellular. So that will the propagation is done to the receptors to adapt to the changes in the body's regulatory environment and maintain the homeostasis. Either there will be upregulation of the receptors, more receptors will come into action, or there will be down-regulation of the receptors, and it will create a balance in the body, and that is balance is the homeostasis. Okay, so these are the functions of the receptors. Clear? Receptors are everyone. Clear? Mostly they are, but not always. Okay, there are some receptors which do not produce. Next. Receptors. I told you site either of the cell surface or inside the cell. Inside the cell it can be cytoplasm, it can be nucleus. Now two definitions, two more things we have to understand. What is affinity and what is intrinsic activity? What is affinity? Anyone? Affinity बोलते क्या होता है? Stand up, say affinity. Can you? Affinity. Affinity is the ability to bind to the receptor that it is designated to bind. Okay. Affinity का क्या होता है? Act at drug, the affinity is more that will act at lower concentration or higher concentration. Okay. So affinity also explains us it is the ability to bind to the receptor. If a drug has high affinity to a receptor, it will work at a lower concentration. And if a drug has low affinity, then high concentration of the drug will be required. Next is the intrinsic activity. It is the capacity to induce a functional change in the receptor. When a drug is coming and it is binding to the receptor, it is initiating a response. That is the capacity to induce a functional change. Either it will give a full change or no change. What will some দিলাম আর কোনো এফেক্ট পেলাম না, তাহলে intrinsic activity is 0. Agonist is going and binding, তাহলে affinity আছে মানে 1 0. So when even if you are giving an agonist কিও bind করে, antagonist কিও bind করে, তাহলে affinity agonist এরও আছে, antagonist এরও আছে, but intrinsic activity agonist এর আছে but antagonist এর নেই। Clear? Intrinsic activity আছে মানে agonist কে bind করলে response দেবে আর affinity was affinity is the binding, so binding to a receptor থাকে। what about the antagonist কি করবে, bind করবে কি করবে না? affinity আছে going and binding, response দিচ্ছে না, তাহলে intrinsic activity কি হবে 0. now agonist and antagonist বললাম, what is the partial agonist? partial agonist এর কি আছে, affinity আছে না নেই? affinity আছে but response intrinsic activity partial মানে in between 0 and 1, 0 থেকে বেশি 1 থেকে কম। okay, partial agonist is going and binding to a receptor, binding ability কি আছে তাহলে affinity কি আছে। okay, intrinsic activity, it will not produce the same effect as that of the agonist, ওইটার নাম কি partial agonist, response দেবে but not like the agonist, full response দেবে। তাহলে antagonist 0 ছিল, 0 থেকে বেশি, agonist 1 ছিল, 1 থেকে কম, তাহলে intrinsic activity কি হলো more than 0। এবারে inverse agonist, affinity আছে না নেই? affinity inverse agonist আছে, it will go and effect কি দেবে কি দেবে না? inverse agonist you stand up বল না, তাহলে inverse agonist কেন বলছি, antagonist তো বলতে পারতাম। so
opposite result of the desired function.
and যদি response দিত না তাহলে antagonist বলতাম, তাহলে inverse কেন?
Opposite result of the desired function.
তাহলে intrinsic response দেবে but it will give a response that is required is an agonist not an antagonist, and just because it is giving the response opposite to that of the agonist, so it is an inverse agonist। clear? তাহলে কি আছে, agonist, antagonist, partial agonist আর inverse agonist সবাই কি bind করছে, capacity to go and bind is the ability to bind, that is the affinity, তাহলে সবার affinity আছে। এবারে inverse intrinsic activity agonist এর 1, antagonist এর 0, inverse agonist এর -1 আর partial agonist এর more than 0 less than 1। clear? now this reaction was given by Clark যে ড্রাগ আর রিসেপ্টর যখন বাইন্ড করে তাহলে একটা ড্রাগ রিসেপ্টর কমপ্লেক্স তৈরি হয়। on the basis of that কি হয়, there are changes, তারপরে এই চেঞ্জেস দিয়ে কি হয়, তারপর রেসপন্স। so this drug receptor complex, it will undergo very transducer mechanism, অনেক চেঞ্জেস। those mechanisms are transducer mechanism, depending on that the effect is good। okay, so drug receptor will combine করলে কি হবে, drug receptor complex, this undergoes many changes and through various transducer mechanisms the response is good। next, now some terms I have already explained coming selectively and binding to that ion channel, in response to which the ion channel is opening and there is movement of the ions. So that is ligand-gated ion. So ligand is any molecule that is selectively to the receptor site. Agonist, both affinity and maximum intrinsic activity present. Antagonist, affinity present but intrinsic activity zero. Partial agonist, affinity is present but some maximum intrinsic activity. Inverse agonist, affinity is present but inverse agonist intrinsic activity negative minus. Next, now types of receptor families. Receptor people produce its effect, initiate the response, depending on which we have classified the receptors into 5 groups. These are ligand-gated, ion channels, deep protein coupled receptors, transmembrane enzyme receptors, transmembrane JAK-STAT binding receptors, and receptor regulating gene expression. Gene expression माने क्या होता है? Nucleus. Here I have already told you can be either transmembrane, ligand-gated ion channel होता है, transmembrane, deep protein होता है, transmembrane माने outer inner में खाते हैं, outer cytoplasm खाते हैं, transmembrane enzyme already name is same होता है, transmembrane JAK-STAT होता है, out of these 5 আমরা 4 out of these is transmembrane and receptor regulating gene expression, that is efficacy. Okay, so all 4 are comfortable. What are the on the basis of on what basis have we divided the receptors into subfamilies? Receptors के हम अनेक group subgroup से divide करते हैं. Subtypes of the receptors. On what basis? Drug action and specific agonist কিছু drug কোথাও কাজ করছে, কোথাও কাজ করছে না. So on the basis of that, we have divided the receptors into like nicotinic receptors, muscarinic receptors. Adrenergic system की बोलेंगे alpha receptors, beta receptors. On the specific agonist and antagonist are on the mechanism of action response they are giving are distribution. কোনটা হার্টে আছে, কোনটা লাংসে আছে, কোনটা টিআইতে আছে, depending upon the distribution, depending upon the molecular cloning, molecular structure basis receptors के আমরা divide করেছি. So these are various ways in which we have divided the receptors. Distribution, specific agonist, antagonist, the molecular cloning, ABCC receptors. Now this part will be covered next. So a short recap of the class. Pharmacodynamics, we have seen the definition. What are you saying? Study of biological and physiological effects of drugs on the body or on the microorganisms or parasites within or on the body and the mechanism of the drug action and the relationship between the drug and transmission. Definition clear? Everyone? Next slide. Principles of drug action. What 5 principles the drug can act? That is stimulation, depression, replacement, cytotoxic and irritation. Next. Mechanism of action, 2 broad classifications. It is non-receptor mediated, receptor mediated. Now, non-receptor mediated it is by the physical action, chemical action, to enzyme, ion channels, to antibody production, and to transporters. Next, physical action examples: osmosis, absorption, emulsions, radioactivity. Chemical action, we have seen chemical agents and antacids. Next. Enzyme mediated, we have seen either it will increase or decrease the rate of enzyme. Enzyme stimulation होते पारे, enzyme inhibition होते पारे. Inhibition we have seen competitive, non-competitive, competitive है substrate and inhibitor एक ही साइड में करते हैं, non-competitive है अलग-अलग तरह के competitive again of two types, equilibrium type, non-equilibrium type. Equilibrium type means if you increase the substrate concentration, it will go and bind inhibitor and response of the substrate will get. Non-equilibrium type की होता है. Even if you go on increasing, you will not get the response because inhibitor ये create a strong covalent bond to the covalent. Okay? So that is the non-equilibrium type. Next. These are the examples. I have already told you that is the substrate binding site and inhibitor binding site. That case is different. In this case it is same. Next. Ion channels three types. কি করে কাজ করে? Either ligand or G protein or voltage oxide. Either next. Receptors. I have told you the definition of receptors. Receptor is a macromolecule. Either transparent cell or inside the cell. It will recognize the signal and it will initiate the response, but itself it has no effect. No other function. Okay. These are the functions of receptor. Next. Polarization cell surface and it is the nucleus. I have told you the definition of affinity, intrinsic affinity. Affinity মানে সবাই থাকে, whether it is agonist, antagonist, reverse agonist or in its partial agonist. সবার intrinsic activity থাকবে কি থাকবে না? থাকবে না depending on what it is. This definition was given by the class theory, drug response, drug-receptor complex response. On the basis of the transducer mechanism, if it changes occurring, the response will get. Okay? Next. So, some terms like ligand, agonist, affinity থাকবে আর intrinsic activity থাকবে maximum, that is 1. Antagonist affinity থাকবে, intrinsic activity 0. Partial agonist affinity থাকবে, intrinsic activity it will be more than 0 but less than 1. Inverse agonist it will have affinity but intrinsic activity will be negative. Next. Receptor types or receptor families. Receptors we have seen where it is located either on the transcription side on the basis of their functions, how receptors are initiating the response. They are classified into 5 groups. They are ligand-mediated, G protein coupled receptors, transmembrane enzyme receptors, and transmembrane JAK-STAT binding receptors. These are all transmembrane. The receptor regulating the cleaving specialist intracellular. Okay, till now we have done so far. Next day, the explanations are dose, effect, response.